Thanks a lot. Goodness, this is really loud. I'm going to be talking to you guys about some of the computer vision challenges that we're facing in our drug discovery activities at GlaxoSmithKline over in the UK. And this is going to be a little bit of a history talk for some of you guys. We've heard during this conference a lot of the type of stuff we can, type of work we can do with deep learning for imaging. We do work on deep learning applications internally at GSK. A lot of the methods I will be showing here will be stemming from more classical computer vision approaches. A little bit of background about myself. I came into the field rather late. I did my PhD in astrophysics at the University of Cambridge. I then did what I refer to as an academic random walk into the Institute of Molecular and Cell Biology in Singapore. And 2 years ago, I joined the GSK R&D hub in Stevenage as a data scientist, whatever this may mean. So throughout that, my kind of leading topic was imaging. So could I have a show of hand vote who believes that on the left you have telescope data? Go. Who believes on the left we have microscope data? Good job, guys. So you can see, even though the changing linear scale here is about 23 orders of magnitude, some of the structures we deal with tend to be very similar to each other. And all of the activities that we carry out as part of drug discovery and all the need to improve on the quality of data analysis and so on is really stemming from the fact that drug discovery is a hugely expensive business. And this slide gives you some of the stats across the industry. It typically takes 7 million hours of man work and 7,000 experiments, 400 researchers to get one medicine to the market. On average, getting that medicine, a novel treatment to the market, costs $2.6 billion. And this is mostly because 90% of the medicines that get to clinical trials fail. And they do not fail because they don't work at all. They fail because they don't work as well as they do in animal models, and they do not beat the treatments we already have on the markets. You might perceive drug treatments, you might have heard of the placebo trials. You don't really do that for serious conditions because it would be unethical. You always have to beat the control group, which is on the best treatment available on the market. So I'm going to be focusing on the discovery, on the preclinical stages of the drug discovery process, which goes from the initial selection of the biological target you are after, which is connected to the disease you've chosen to tackle. But the first example I'm going to give you comes from the lead discovery phase. And this is once we have selected a condition to treat, we have selected a drug target that we want to treat. We are looking for drug molecules that actually will do that for us. And whilst there is a lot of effort going into predictive modeling for those intents and purposes, and some of the newcomers to the pharma market are actually, like Benevolent AI, based in London, are focusing all of their strategy on in silico drug design. And I believe there was another talk I wish I could catch that was speaking on that specific problem. Still, a vast majority of work carried out is based on a huge amount of screening of existing compounds that we know how we can make and seeing whether they fix any of the disease we may be interested in. So if you think about it, it's a huge selection process, and you want to increase and boost the quality of the filtering that you are doing. And those assays or sets of experiments get progressively more expensive and more complicated as you go on. A lot of that analysis relies on what's called high-content profiling, where your readout from the experiment is images. You will tag different parts of biology with fluorescent dyes, and you will therefore have labeled images corresponding to biology structures. There was a great talk by David last yesterday that spoke about the Kaggle challenge on nucleus detection. The reason why this is a very important and relevant problem is it's the basis of how all of the commercial pipelines available on the market are constructed. First, you find this, the cell nucleus, because it always has one. And then from that, you can grow either via Voronoi tessellation or via watershed to some other marker. You can then find your cell and be able to constrain any analytics you do to a relevant region of interest that corresponds to a biological entity. And we are after characterization of a biologically relevant population, really. So overall, you quantify the phenotypes using some sort of imaging features. There's a bunch of software that already does it on the market. And then you want to discover how do your chemical compounds modify the imaging phenotype. And the real motivation about doing some more advanced analytics is that even though everybody talks about high-content profiling, not many people actually do it. So this is from a review paper back in 2014, which looked at how many papers have high-content screening, the HCS, in the title. And then on the x-axis, you've got how many features of data they actually look at in their analysis. So more than 80% of papers at that point were actually only looking at a single feature of the data, or maybe two features of the data. Arguably, this is not really high content, at the least. So the specific biological context I'm going to be talking about here is treatment of COPD, which is a pulmonary disease. It is slowly lethal. There is no treatment at the moment, and GSK is a company that is leading in the field of respiratory treatments. And this is the disease to the treatment of which we're committing a large amount of resources. And basically, it has been demonstrated that in this specific disease, as well as in the others, what you have is you've got problems with the activity, with the phagocytosis. So with the ability of macrophages, your immune system defense cells, to eat up the bacteria that enter your lungs when you breathe. And this leads to build-up of mucus and sort of slowly growing blockage of your lungs. You physically stop being able to clear your lungs. You stop being able to breathe. So our biologists developed this rather interesting assay where we've got the population of macrophages in vitro. We introduce one of the dyes into the macrophages. We introduce a pH-activated dye that only becomes fluorescent in acidic environment. And we introduce that inside bacteria. So when the bacteria get hoovered up and eaten, digested by the macrophages, this is when you get the fluorescent readout. So you have a direct measure of the activity you're interested in, of phagocytosis. Now, of course, there was a bit of a challenge in terms of the data analysis. They were looking at very simplistic readouts, and they weren't really interested in doing a lot of the... They weren't getting out as much as they could. They were looking at sort of population level aggregates as opposed to at exploring how the populations behaved. I'm not going to go into too many details about that. But first tip in terms of having a computer vision project, check if there isn't an already available solution out there. And if you guys do need to work in the biology space, Cell Profiler is an excellent package made by people at the Broad Institute in the Ann Carpenter lab. This is a software that is optimized for kind of classical analysis of cellular-based assays. It's got a GUI that mirrors some of the commercial software produced by the microscope manufacturers, so it's immediately familiar to your biology friend, because you need a biology friend when you do any sort of work like that. And it does a good job of extracting all of the classical computer vision features that we're going to cover later in the talk. And it can be launched from the command line, which means we can deploy that on a server. And this is a very naive bit of code that uses the queue and the threading libraries. So I've got my worker defined. The worker literally grabs the worker from the queue, sets item is what gets passed. CP thread will launch the process, and this is just where Python and cellprofiler.py happen to live on the server I was using. And you pass it, crucially, you've got that args pipeline that will point to your relevant config file that your biologist created for you. And then your queue will, for every worker for every worker thread that you have initiated, it will assign a worker. And then for item and crawler, this is just a simple code that traverses the directory structure, identifies where the data lives, does a little bit of cleanup, and gives you the it gives you the directory structures that you need to pass. Cool. And what we did with that is essentially we looked at the underlying morphologically distinct populations of macrophages. So over here you can see you've got a bunch of them that are kind of roundish and a bunch of them that are thin and spindly. Those are functionally different macrophages. They do different things in for your immune system. We've managed to have thanks to those single cell level features, we've trained a classifier, straightforward SVM, that predicts whether the individual cell comes from a healthy or a diseased population. And then what you see on the bottom here, this is a mock-up of a layout of what's called a well plate. So those are actually your kind of parallelized biology systems where you have a plastic plate about this big. It's got 96 wells on it. It's got transparent bottom so you can do imaging. And what's plotted in the intensity is the fraction of cells that were given the healthy label. And what is reassuring is that this actually comes from a plate that came from a disease model after it has been subjected to our candidate compounds. So in a way, you can measure, using your SVM classifier, you can measure the fraction of the cells that appear to have reverted to a healthy phenotype, which is what you want, which is what your medicine wants to do. It's to revert back to the original state, pre-disease. So this is challenge number one. Challenge number two was a different assay, which actually broke most of the commercially available software, because this is a collagen deposition assay. And the reason why it doesn't work classically is rooted in the biology. So over here you have collagen fibers, which is the stuff that makes your hair, deposited by the cells inside the well. So all you have here is the fluorescent collagen. Now, the problem so collagen forms your hair, but it also forms any of the scars you may have in your body. And when you have scar tissue created in places where it shouldn't be, that's a condition called fibrosis. There's many different types of fibrosis depending where it is, but this is, if you have it in a crucial system, fibrosis in the lungs, fibrosis in the heart, that will kill you. We don't want that. So we want to understand how can we modify this process. And so we need a good in vitro model, and we need to be able to quantify that. The reason it doesn't work with the classical approaches I mentioned earlier is over here you do not really have a direct connection between the individual cells and the collagen fibers, because the collagen fibers grow outside of the cells. There's no ability to connect them directly, saying, oh, this is clearly this particular cell that produced this collagen fiber. No, it's an emergent structure made by several adjacent cells. So we want, at this point, to do a analysis pipeline that just looks at this whole well overall. And crucially, we want to, as a kind of challenge we set ourselves, we want to completely ignore the intensity, because all they were doing was, how much collagen do I have? How much signal? which is a good first take, but we wanted to see if we can do better. We wanted to see if the texture of the collagen tells us something about that. So this is a peek into the feature extraction function that is reliant on the Mahautas package by Luis Pedro Coelho, which is a really good NumPy optimized, for those who were on one of the last talks yesterday. It's a really good, really quick implementation of plenty of the standard computer vision features. I can highly recommend it. As you can see, it is really trivial to implement, and I pretty much have a single line of code gets all of your Haralick features. Single line of code gives you PF-TAS features and the LBP features as well, which is great. What on earth are those things? And this is where the history portion of the talk commences. Haralick texture features are pretty much the first foray into proper quantification of image textures, all the way back from 1973. And it's basically measures of things such as correlation, variance, and entropy defined with response to this entity called the co-occurrence matrix. The co-occurrence matrix basically tells you if you have p levels of intensity in your image, it tells you how often they occur next to each other for any combination of p for a given definition of adjacency. So the way you normally deal with those is you report the Haralick means, which basically get measured on the 4 adjacency directions down, sideways, and the 2 diagonals. Slightly newer, I say, 2007, are parameter-free threshold adjacency statistics. So crucially, the key difference is Haralick means are independent of any thresholding you might have. This was purposefully made for biological applications, for something where you have an entity tagged with a fluorescent probe. So thresholding becomes a very natural thing to do. So over here you do some sort of thresholding, and like everybody in biology, you probably default to OTSU. And then you look at what you have on the right. For every pixel that has a signal in it, you look at the sort of 3 by 3 pixel set, and you look at this individual pattern, and then you basically have a frequency distribution of all of the potential patterns of pixels, sort of one encoded, if you wish. And if you haven't heard of OTSU thresholding and how it works, and hopefully this will play. Famous last words. No. Okay, so essentially the OTSU thresholding deals by splitting your image into the foreground and the background class, and you want to minimize the intra-class variances, which OTSU has shown in 1979 is identical to maximizing the inter-class variance between those two. It's a very efficient algorithm that essentially scans, it's an exhaustive search, scans through all of the intensity levels and finds the thresholding that is optimal for that purpose. And it runs very quickly, which is always a great feature. Okay. And this should really... Okay. Fantastic. Yay for Windows and encryption. Cool. So whilst my laptop decides whether it wants to keep working or not, just a quick introduction. The final set of features is the local binary patterns, and the way you deal with those is you have a set of 8 neighbors that you define in the 8 cardinal directions, and then if your neighbor is lower than the central pixel, you give it a 1. You give it a 0. If it's high, you give it a 1. And then you concatenate all of that, and you have a binary digit, an 8-digit binary number that you then create a create a histogram of, well, you essentially select the rotationally invariant ones. Cool. So it's just my PowerPoint that has crashed, so it's no biggie. Have you got the slides up? Cool. Never trust your laptop. Always have a backup. Great. I'll just dump that here. What would be really funny if this crashes on the same slide transition? Okay, and the video even works. There you go. So this is the scanning through the different thresholding space that the OTSO does, and you can see the interclass variance, which you're trying to maximize, being computed at every step. And once this goes to the end, you'll see the maximum will result in your split to the foreground and the background, and certain applications will allow you to do multiple OTSO thresholds subsequently if you believe that your foreground should be further divided. Hey! Skip ahead because getting close to the end. So essentially, when we looked at the feature space, we had two controls. There's some red points, some green points. Green points are the ones where collagen gets deposited, but no treatment is present, so no chemical compound that modifies the process. Red ones, we did not introduce TGF-beta, which is a biological enzyme called the growth factor, which actually stimulates the production of collagen. And we see that the purple points, which are the compounds, actually have a very significant scatter in this space. However, what plenty of pharmaceutical assays are designed to do is they are designed to give you a response with respect to concentration. You want your drug to have a stronger effect when you put more of it into the system. So this is what we looked at. We looked at the compound tracks through that feature space. Sort of how do those behave with increased concentration? And we essentially saw that it's pretty much reliant on the first principle component. And when we just extracted the first principle component, we compared that to some of our other methods of quantization on the bottom, and you could see that it was rather nicely behaved. Biologists were happy, and they decided to stop this project because they were happy with the output at this point. As a side effect, we were able to give them an outlier artifact direction for free. So some of those outliers in the second principal component were cases where you have hugely bright localized features, and those are not your collagen. This is just a piece of junk that happened to contaminate your sample. And, if you're doing a manual analysis, you would manually remove them. If you are screening through 70,000 compounds, you're probably not going to be willing to do that. Just very quickly, there's a bunch of stuff, a bunch of analysis that happens in the preclinical studies, and this is a hugely entertaining system. So on the left side here, you see a demo of the OptoSelect technology by the Beacon, by Berkeley Lights company. So what you're seeing is an individual cell being rolled around on the surface of the chip by illuminating the area around it. The way that works is they have a localized photovoltaic effect, and it's the electric field that gets stimulated locally that repels your cell. Now, what that lets them do is in the center here, you have a layout of a chip that contains something like 1,750 nanopens, and you can use those light gates to grab individual cells that you flush through the system, put them into those individual pens, and they will happily grow over a couple of days. Why we are interested in this technology is for manufacturing purposes. So there's, broadly speaking, two types of drugs. There's your small molecules, which are chemically synthesized. There's also antibody treatments. And we cannot synthesize those, we need the proper biological mechanisms. So those are made in production cell lines in large bioreactors. We want to have them as efficient as possible. And for that purpose, we want to select the most productive clones. So just really quickly. Or not. So the purpose of that was really to we did a small project I'm going to skim over that was basically tasked with augmentation of some of the capabilities of the proprietary software that was delivered with this machine. We did a couple of things. Again, we did artifact detection. We did some straightforward machine learning classifiers. Or we did all of the groundwork for machine learning to be deployed and be incorporated into the decision-making process on which cells to export as soon as we get the proper training data, which is, in this case, features related to production, and the scale-up for that takes weeks and months. So we do not quite have a training set to train proper machine learning models, but some of the toy ones we have with some. Oh, there you go. Oh, now you work. So we have some toy models that basically seem to predict how well behaved and how smooth the antibody distribution within those nanopens is. Because if you see a lot of this clumping, that could be an early onset or early marker of aggregation, which is a huge production issue. This is mainly why antibody systems do not, antibody cell lines do not really work. So that's all. Thank you very much. And if you have any questions about the work we do,
one short question.